there are some unique situations in neuroendocrine tumors, and one of them is when you have a husband and wife team. And even more important than that is if the husband is an endocrinologist and the wife is a pediatric hematologist, oncologist, you couldn't ask for a better combination than that. Tom and Sue Sodorisio and I go back many, many years, almost to the age of the first discovery of the first gastrointestinal hormone in Secreton in 1902, and we've been together over many, many years, seeing all the different phases of evolution of gastrointestinal hormones. I want to say about Tom, he was very instrumental in helping me make my career when I came from South Africa to the United States, and he was on a study section and helped them promote the type of granting we were seeking at that point in time was for the diagnosis and management of neuroendocrine tumors. And I have a lifetime of debt to the man who I call the gentleman of science to be able to do that. Now, as regards to Sue, I loved it when she became involved in the world of gastrointestinal hormones and neuroendocrine tumors. She pioneered the understanding of the receptors and the different receptor subtypes for somatostatin. She took a gastrointestinal hormone, vasoactive intestinal polypeptide, and began to look for new targets and worked out a whole new action of vasoactive intestinal polypeptide on leukocytes, circulating leukocytes, and developed the relationship of the gastrointestinal tract and its hormones with immune-mediated mechanisms. She furthered the notion that there could be small molecules that were targets and receptors for these peptides, and that then could be used for tracing and identifying the tumors, something she has assiduously and steadily and steadfastly adhered to and gone further with that. Not only that, is when she moved from Ohio State to Iowa to become chairperson of the Department of Pediatric Hematology and Oncology, she didn't drop the side of her work, and she pursued that despite having an active career in running a department, and began to champion the cause of children with medulloblastomas, also looking at children with neuroendocrine tumors, trying to bring Y90 into the arena of the clinical trial to give children access to the state-of-the-art therapy for this condition. So I have indeed the great pleasure to introduce her to you, to talk to you about the genetic signature of neuroendocrine tumors. And Sue, thank you for doing that for us.
Thank you very much, Maureen. Wonderful venue for this meeting. And I've said to Jim already that I have been very inspired by his opening remarks to tell us all that we need to put into this a lot of effort in order to be able to go home and change our lives. And I hope that today I'll be able not to give you a very erudite talk about what's going to happen 20 years from now, but rather to, because I'm not a geneticist myself, be able to tell you, give you an understanding, a little bit of understanding of what your genes are. This is kind of the talk that I would give to college students or graduate students if I were going to tell them about the genetics of neuroendocrine tumors. So it's not 20 years from now, it's kind of now, and to help you actually speak with your doctor and understand what your doctor is saying to you. Now we've talked, I tried to make a list actually over yesterday and up until today of how many different genes we've talked about. And I'm up past 50 now, and if we could all understand how those 50 genes interacted together and know that we have another 45,000 in our body and in every cell, that's what it's going to take for us to get to be able to understand what everybody's been saying, is that each one of us who has a neuroendocrine tumor has a very unique tumor. So if we just understood these genes, we've talked about genes that can actually be biomarkers. Sometimes we use the peptide genes for biomarkers. We can use them as targets for therapy. That's the way we use a lot of the receptors, as targets for imaging and for therapy. And most importantly, if we could understand all of these genes that regulate how a cell divides, and where we could target new therapies to be able to hit which pathway in whose tumor. We really, that's what it's going to take for us to get where Dr. Modlin wants us to be in the next 10 years. I want to give you today some understanding of what chromosomes and genes are and how they all fit together. I'd like to show you what a mutation means and give you some idea of what we're talking about when we say there's a mutation in a gene, and demonstrate how we use these genes in cancer diagnosis. You already know a lot about it. We just don't think about it as genes when we talk about what was your gastrin level in the last talk. When we've talked about Ki-67, we just don't think about them as genes. And what we have to do is to be able to put all of these together to understand the makeup of a cell. And that's what's going to help us predict what the neuroendocrine tumor strategies are going to be in the future. Well, Dr. Urbain said it last night, almost word for word, what's already here on my slide. He said genes are pieces of DNA. And I just told you we have about 40,000 of them in every cell. They're also blueprints for proteins. Dr. Urbain said that almost exactly word for word last night also. So you are, probably all of us are most familiar with the interaction of two genes, somatostatin, the peptide, with its receptor, the somatostatin receptor. I'm going to try to show you a little bit today and have a little fun with you regarding how genes work together and how we can predict what we can predict now and what it's going to take for us to predict what Dr. Modlin and Dr. Urbain talked about to be able to give a genetic signature not to one tumor but to every one of us. So your chromosomes are actually the storage for your genes. Your chromosomes are in the nucleus of every cell. They actually come in pairs. So every one of us has, in every single cell in our body, 23 pairs of chromosomes. So are there any chromosomes in this picture of how many chromosomes there are in every cell? You see the pairs, and you can see that they're numbered from 1 to 23. Let's go all the way to the 23 and talk about, okay, there are men and women in this room, and I'm one of the ones with 2 X's, and then you have the X and Y. And you all know some genetic diseases. You know we can have too many chromosomes, and the one that's illustrated right here is a child. I work with them a lot, the children with trisomy 23, or we call it Down syndrome. I always say I think they have an extra copy of the happy gene, but along with which I think must be located on chromosome 21. We haven't found it yet, but along with that, unfortunately, I see them because they have a greater risk for cancer. So we have good things and bad things on every gene. You can have too much of a good thing, and a chromosome 21 is a good thing, but you can certainly have too much of it and end up with a disease. Now, does one chromosome make a difference? We talked a little bit about the X and the Y chromosome, and yes, it does. It tells us the difference between males and females to begin with. But there is a tumor suppressor gene that's on the X chromosome that turns out to be very important in neuroendocrine tumors. On the Y chromosome, if you don't have the testis-specific protein gene, then you actually become a woman. Now, I think there's some other important genes on the X chromosome. My son actually thinks my son-in-law thinks that I have 3 or 4 extra copies of this STF gene. He calls it the slip, trip, and fall gene, and he's sure that I gave 2 extra copies to my daughter. Now, I think that I always thought when my teenage boys were growing up that there must be a toilet seat gene on that X chromosome, and now when I try to talk to Odo at night, I think he is expressing that channel changing gene and just doesn't hear me at all. But there are some chromosomes, actually, not just genes, that have given us hints about neuroendocrine tumors. One is in midgut carcinoids, and this is a chromosome 18, and in 50, when the studies have been done, 50 to 80%, almost 90% of midgut carcinoid tumor cells have something missing from chromosome 18. Now, when we look then for genes that might be important in midgut carcinoids, we have, by looking at the chromosomes, we have a good place to look. The problem is that if you look at the 18th chromosome, there are lots and lots of genes, not just one or two on chromosome 18. So we haven't done our work yet when we say, okay, we know if you have something missing from chromosome 18, you're at more risk for a midgut carcinoid, but there might be, 5,000 genes there. So we're not home yet. Similarly, with other gastroenteropancreatic tumors, there are losses on the 9th chromosome. We heard Dr. Asa say very first day that the 11th chromosome was very important for the MEN1 gene, and that it's important in lung cancers as well as in pancreatic neuroendocrine tumors. So you see, and here's that X chromosome. Now the important thing about this X chromosome loss you see we have 3 tumors there. The GI tumors, the pancreatic tumors, and the colon tumors. What unites these are the tumors that are what we call the poorly differentiated neuroendocrine tumors. So here's a gene that has to do with likelihood of an area of the chromosome that has a likelihood to do with whether this tumor is going to be very aggressive and whether it's going to metastasize. But again, we know where it's located. It's on chromosome X, but there are lots and lots of genes on chromosome X, and we don't know which one is important. Look at, though, the amount of the percentage of tumors that are poorly differentiated that have something wrong on the X chromosome. And we heard yesterday, another gene that I didn't have on the slide, beta-catenin, is a very important gene in all of these tumors as well. We know that beta-catenin is important in cell cycle regulation. That is, when a cell does decide to divide, and what are the brakes that you put on this gene, but we don't know all of the answers to those pathways, let alone be able to decide a drug that's going to target that area yet. So how many genes are there on one chromosome? Well, it depends on the size of the chromosome, and if you remember the picture of the chromosomes, they were actually numbered according to their size. So number 1 is the biggest chromosome, and the Y chromosome is actually the smallest one, and we think it probably has 200 to 500 genes on it. The X chromosome is bigger. Remember that picture, and it was quite a bit bigger, has about 2,000 genes on it, and the biggest chromosome, chromosome 1, has more than 5,000 genes on it. So you see why there may be a little problem just to say, well, we thought we were making a lot of progress when we said, well, if we just knew what was going on the 18th chromosome, we would know how to diagnose and treat these tumors, but now we're down to 5,000 genes, and that doesn't even talk about how they interact with the genes on the other 22 chromosomes. So I thought I'd give you a little lesson in how our genes put together and help you understand. I think I heard at least once yesterday genotype versus phenotype. So how many of you in the room have brown eyes? There are quite a few of you who have brown eyes. Now, brown eyes are very common. How many of you have blue eyes? And I see almost as many looking at blue eyes, but blue eyes are actually more rare than brown eyes are. So we always thought that right after we discovered, Dr. McClintock got the Nobel Prize for looking at the genes in corn, and that was pretty simple, that eye color was the next simplest kind of gene pattern that you could look at. And someone discovered that there was a gene, which was aptly named eye color, so EYCL for eye color. How does this control your eye? Well, it's on the 15th chromosome, so you see now down there, it's again with the X chromosome and the Y chromosome, and then on the 15th chromosome, there are genes that control your eye color. Now, let's say, I think I saw Odo raise his hand when I asked about brown eyes. And I should have raised my hand when I talked about blue eyes, because I have blue eyes. So let's just look here. Odo has brown eyes. Remember, we have 2 chromosomes, so there are 2 copies, so he has 2 copies of that gene. And I have blue eyes, so I have 2 copies of the blue. Now, the red lines connect our 3 kids, and one of our kids, our first son, of course, because he got 1 from Odo and 1 from me, had to have brown and blue eyes. But that isn't quite as dark as his eye. If you look really closely, you can see there's a little tiny tint of blue in that square representing Joel. Now, our daughter's the same way. She had to get the same thing. She got 1 copy from Odo and 1 from me, and has brown eyes. But there looks like there's a little hint of blue there as well. And our son's the same way. It couldn't have been any other way, right? Because they got 1 copy from each of us, and we were the purebred dogs the other way, because he's Italian and I'm German. So now our kids are the mixed, and I felt kind of cheated because none of my kids had blue eyes. So is there any way that my grandkids can have blue eyes? So our first son married a young woman who had blue eyes. And can you go backwards for me? And you can see on the left here, then if they were to have 2 kids, we would predict that they would have 2 children with blue eyes. And the others would have brown eyes, but you can't tell just by looking at the brown eyes whether they can have children with blue eyes, right? So it's a little more complicated than just say, we're going to mix and match. So now here's our daughter, married a man with brown eyes, and of course they don't have any children with blue eyes, but their children would be able to have, depending on what the mix was with their spouse, they would be able to have children with blue eyes. And finally, if our youngest son married another person with brown eyes, but they both have a little touch of blue in them, then they could end up with a child who had blue eyes. Well, that seems pretty easy, right? But what we now know is there's not just one. Remember that said eye color, gene number 3. So even eye color is much more complicated. Now, there's some people who didn't raise their hands when we were talking about what color eyes you had. So, do you people have hazel eyes? Ah, see? Or anybody here with green eyes? Another one. So there must be more than just these 2 genes. Here's the blue eyes. We already talked about that one. How about anybody with gray eyes? 1 person back here with gray eyes. And does anybody know a child or a person who has red eyes? So if you have a complete lack of eye color, so now we know there are 3 eye color genes that allow us to have this big variety. And if you lack all of those 3 genes, you're the person who has red eyes, because all you see when you look in their eyes is the blood vessels reflecting the light. And the red blood gives you the red color. So imagine then, if it's just this complicated to determine eye color, how complicated it must be to determine what makes a tumor and how we're going to treat it. Now, broken down to the simplest explanation, there was a person who actually won the Nobel Prize for this. There have been quite a few Nobel Prizes for genetics, but this is a Nobel Prize for understanding what it takes to cause a tumor. So we'll go back to the idea that on some of these genes, let's say this is chromosome 18 that we're looking at here. You have 2 copies of chromosome 18, and on one chromosome 18, in your neuroendocrine tumor, you have lost a piece of that chromosome. And then someone asked a question yesterday about benzene. And we know benzene can be an environmental toxin that can actually cause mutations in genes. So let's say the benzene caused a mutation, and it just happened, it's random as far as we know now, that it just randomly made a mutation in that second gene. So now you've got 2 genes, you've got a gene on each of your 18th chromosome that have a mutation. And that's the simplest thing it takes to make a tumor. We can actually now do this in the laboratory. You can take the cells from a person who has a mutation in one gene in their chromosomes, and in the laboratory expose those to environmental chemicals and end up with a mutation in the second gene, and it'll become a cancerous cell in the laboratory. So what's the scientific name for these hits that Knudsen thought must be causing the cancer? The hits are actually mutations. They might be just a change in one amino acid. That change in one amino acid of the protein can be looked back at in the DNA, and in this piece of DNA, it might be a piece of DNA that's about 1,000 nucleotide base pairs long, and you may have one change in one of those 1,000 nucleotides in the DNA and result in a mutation in the actual protein. Can make it work more. That's why you can have, even without having too many chromosomes, you can have too much activity of a gene if you have a mutation in the gene that makes it more active. Similarly, you can make it, if you have a tumor suppressor, you want those genes to work, and we now know that mutations in tumor suppressor genes can make them not work. And in some cases, either a mutation or knocking out a piece of a chromosome can actually completely knock out the function of a gene. So what are some of those genes that we know about in neuroendocrine tumors? You've heard of all of these. And the mutations that happen have been substantiated in all of these genes. So if you have one copy of the RET proto-oncogene, and then you are in that family, you can see that the transmission of that gene is going to be very similar to the transmission of the color of your eyes. So you may have a hidden copy of that gene and not have it expressed, and not have it unless you get another hit, not have a cancer unless you get another hit. So you can talk about the BRCA gene, which is in my family, and you can say, we have the prediction that anybody who has that mutation is going to have breast cancer, but you can't say 100%. You can say by age 50, there's a 50% chance. You can say by age 70, there's a 70% chance, but you never can say there's a 100% chance because you need that second hit to be able to cause that cancer. So these are names of genes that you've seen, particularly if you know you have this gene. You also know that you're at greater risk for a neuroendocrine tumor, and that your doctor should know that from the time that, and your children's doctors should know that from the time you first take your child to the doctor. So how do you know whether you should ask your doctor if you need genetic testing? Well, we're certainly not at the point where we're sequencing everyone's genome yet. We really don't know what to do with all the information yet. But if you have a first-degree relative, and that means if you have a parent, a sister or brother, or a child who has a tumor, then you need to have genetic testing of all of your family. So when we found out that my mother had BRCA2 gene mutation, 11 brothers and sisters, we all got genotyped, and we're down now to, but what that helps you with is, you know, my kids don't need to be tested because I didn't end up with the mutation. But my brother's 3 daughters all got the mutation. So it helps you understand. And for those of you who have already some knowledge of what mutation is in your tumor, if you have the VHL gene in your family, if you have the RET proto-oncogene in your family, you know you need to have genetic testing, not just for yourself, but for your siblings and your kids. So if you have any of the traits, often, particularly for neurofibromatosis type 1, we do not do genetic sequencing, even though we know the exact sequence of that gene. We don't sequence actually. So that's how far away we are. Unless we see a child who has the NF1 and has some suspicion of a tumor, then we would sequence that gene. But we were talking today about doing our whole genome for $6,000 in 24 hours, and actually the reality is that to test the neurofibromatosis gene and sequence it takes 2 weeks and costs $7,000 right now, and that's just one gene. So I think the other people who are going to get sequenced are those people who right now have $100,000 to be able to spend on their whole genome sequencing, and for the rest of us, I think it's a few years away yet. So what? How can? But can we, for those families? Even without sequencing, can we make any predictions? And this is why your family history is so important. There have been some studies by Dr. Hassan, actually, at the MD Anderson, who looked again at the SEER database that we've heard about so much, and looked at what were the risks for gastric carcinoid in women. Actually, she did the study on both sexes, but this is what turned out to be the most significant in the work. If she did find out that for men and women, there did not seem to be any greater risk for these gastric carcinoids if they, depending on whether they smoked or not, or whether they used alcohol or not. What they did find out was that there was some increased risk, and you see under cancer there, it says the increased risk is maybe about 2-fold increase in risk if your family had cancer. If your family had diabetes, and it was much more significant if your family member with the diabetes needed to use insulin. So type 1 diabetes was more of a risk than type 2, because most type 2s, at least in the beginning, don't need to use insulin. But look at this risk. The risk for diabetes by itself was higher than having cancer, and that was about a 6-fold increase in your risk. But if you had a relative who had both cancer and diabetes, you had a 50-fold increase in the risk of having gastric cancer if you were a woman. So there are ways now, although we're talking about individualized medicine, we still need to do some epidemiological studies on all of us as a group to be able to understand these kinds of risks. Now, one of the things that parents ask me a lot is, how do I know what the risk is for my child? And particularly if you have a small intestinal cancer yourself, and then your son or daughter comes up with a bellyache, what's the first thing you think about? Oh my gosh, does my child have a tumor? So there actually is an increased risk for your children if you have a small intestinal tumor. Seems to be the most risk to be able to pass it on to your child. Although again, we know a risk, but we don't know what the genes are. So you can see there's a 10 to 12-fold increase in the risk of your child having a small intestinal tumor if you have one, and about an 8-fold, a little less risk for your brothers and sisters to have a tumor. The risks seem to be much smaller for colon and lung cancer, lung carcinoids, if you have a lung carcinoid. So let's just talk about some of the new genes and some of the old genes that you already have heard mentioned. The lung neuroendocrine tumors, of course, the MEN1 gene is very important in those. We know some of the genes that are important in gastric cancers, in ileal cancers. These are some of the genes that we now are starting to put genes on the chromosomes and starting to narrow down from, well, there's something wrong with my whole 18th chromosome, to what is the actual gene and the gene area that we need to target on those chromosomes. Now you can see the pancreatic NETs, and actually this is, Dr. Modlin has done a lot of this work. He showed you a little bit this morning of how we can now begin to put a pattern together of genes. The problem is now we still don't know what's the relationship between these genes that are in that pattern. And it takes a lot of work in the laboratory to understand, because it's not really always intuitive how these genes work together. We do use genes to diagnose neuroendocrine tumors. You all know about it. I call this diagnosing by one gene at a time, and we might be up to the stage now where we're diagnosing neuroendocrine tumors by 10 genes at a time, but more likely it's one gene, and oftentimes it's one of those peptides that we've talked about so often that gives you a gene product that actually tells you there's a suggestion you might have a neuroendocrine tumor. We can stain genes. You've seen a lot of pictures of cells stained. You've seen the Ki67 stain. You've seen the somatostatin receptor gene stain. You've seen the neuron-specific enolase gene. These are all ways of identifying genes that are important in your cancer. And I think we're going to have the pleasure of hearing this afternoon. We've already seen how we've targeted genes to have what I call molecularly targeted imaging. And I hope we'll hear from Eric this afternoon. This is really molecular therapy. PRRT is actually targeting a gene. And we talked about using more than one gene, or how we're going to be targeting 10 genes at a time. And I think this may be one area in which we'll be able to do that. So your tumor may express the somatostatin receptor, but we've just been able to image the melatonin receptor, And there are tumors, actually, I'm very interested in kids' brain tumors, and kids' brain tumors actually express both of these genes. So I foresee the day when we will be able to be using 2 radio-labeled peptides in one treatment. So you give that cell an extra hit, and we may be able to make our peptide radiotherapy much more effective in this manner. So in summary, I want you to understand that chromosome losses and gene mutations do cause cancers. They're extremely important in neuroendocrine tumor. I think the bad thing is we really don't, we have maybe, we talked about 50 genes I said here, but think about the other 35 to 40,000 that have to interact with these genes that we don't have really out there able to fit into the rest of the puzzle today. So we're just beginning to put that puzzle together. And I think that's why we're just beginning to understand the genetic causes of neuroendocrine tumors. We know actually much more about how to exploit those genes in order to do imaging and to do therapy than we do about understanding what we can do to prevent the tumors. And that's really what I think the promise of the of knowing the whole genome and of us understanding more of how all of these genes work together is that we'll be able to predict, as Dr. Asa talked about, that people with some kinds, one copy of the gene may be able to respond to a drug, and people who have a different copy, which isn't necessarily a mutation, but just have a different form of the gene, sort of like blue eyes and brown eyes. So the people with the blue-eyed tumor may respond to a drug, but the people with the brown-eyed tumor won't be able to. And I think those are some of the ways that we will be able to tailor therapy to a specific person. And I think, as Dr. Urbane said so well last night, and Dr. Modlin talked about this morning again, I think the future of us being able to treat neuroendocrine tumors is to understand these multiple genes and to be able to treat them all at one time. So why should you as a patient learn about some genetics? I think it helps you understand your disease a lot better. By understanding what genes are important in your tumor, even the few that we know about now, even though we don't know about the whole 40,000, the 10 that we do know can help you determine whether you need genetic counseling or not, and whether your children are at risk for having the same disease. It helps you much more converse with your doctors, and as I think everyone has said, multiple people have said, you know much more about it, and I know this from the standpoint of seeing my own patients with other kinds of tumors, that parents often bring me new information, and you will be able to bring your doctor new information. I think it's important when you may sometimes, and it's becoming closer and closer every day, that we're going to need to justify to our insurance companies what our genetic makeup is and what it means for our treatment. I think it's fun, actually, to be able to talk about these genes, and I enjoy teaching my kids about it, and I hope that you will be able to do that as well. And finally, I think that this is going to help you advocate for yourself and for your children.
Sue, thank you very much for that wonderful idea. That was really magnificent.